As usual, we're going to have a mini seminar followed by a discussion of the organization of the working group. I need to remind you that the meeting will be recorded. It's being live streamed and it will be available afterwards. So please be aware of that. Our agenda today has one excellent presentation by Amber Smith. Just to remind you, I'm James Glazier, co-lead of the working group with Reinhard Labenbacher, who'll be taking over after Amber's mini presentation for the business part of the meeting. Jim Sluka is responsible for web administration, and you probably had correspondence with Bruce Shapiro, who's handling that aspect of the organization. If you need to talk to any of us, connect any of us, our emails are available here. And we have a Slack channel. As I keep saying, please use the Slack channel. It's only as good as the people who use it. We welcome your participation in the Slack channel. If there are any issues with it, let us know. Jim Sluka can help out. We have the IMAG MSM wiki page, which needs content. So please contact Jim if you need help working with the wiki page. Without further ado, I'd like to introduce Amber Smith, one of the great people who combines experimental work with computational and mathematical work, who's going to give us an introduction to the issues of bacterial viral co-infections. And I don't want to take any more of your time, Amber, so I'll turn it over to you.
Okay, thank you. And thank you for the invitation to talk today. Get my presentation going. Okay, so as James mentioned, I'm going to talk about modeling viral and bacterial co-infection. I'm mostly going to focus on Influenza-Pneumococcal. So for those of you who might not be familiar with this particular co-infection, bacteria often complicate influenza infections. During seasonal influenza, the prevalence of co-infections is, can be relatively low, but also non-zero. Different studies have estimated somewhere between 2 and 35% of all morbidity and mortality is associated with the secondary bacterial infection. But where we really see this interaction is during pandemic influenza. And some retrospective studies for the 1918 pandemic have estimated that over 95% of the morbidity and mortality from that pandemic was actually due to bacterial co-infections, not just the virus by itself. And that's not to say that the virus was not also extremely important and deadly, but most of the morbidity and mortality was due to the secondary bacterial infections. In the 1957 and 68 pandemics, this was closer to 50 to 70%, and that was also a different virus, or two different viruses that were infecting there compared to the 1918. And more recently, in the 2009 pandemic, this was more of a prospective study, and about 45 to 50% of all the morbidity and mortality was due to secondary bacterial infections. Pneumococcus is one of the primary pathogens that is associated with influenza. It is an important pathogen in and of itself. It's the #1 cause of community-acquired pneumonia. It's a top 5 cause of meningitis in children, and it can cause all kinds of different diseases throughout the body. But for this particular infection, we're mostly interested in the pneumonia aspect. A second really important pathogen is Staph aureus. And that's becoming more prevalent in recent years, in part due to the antibiotic resistance, but also due to, maybe to a lesser extent, that we have pneumococcal vaccines that we've had for the past few decades, although they don't cover every strain. So we, of course, still see quite a bit of pneumococcal infections. So we have the fortunate ability to use a mouse model. The mouse model really recapitulates a lot of the aspects of human disease in terms of the disease course, the immunology, some of the interactions that we might find. Everything seems to follow what happens in humans, although of course humans are extremely difficult to sample, especially when they are very sick with 2 different pathogens. But in the mouse model, there have been several things that have been discovered. And so this first piece of data goes back to one of the original characterizations of the mouse model, where they determined that survival is really dependent on the order of the infection. To the extent that if you actually have the bacteria first, depending on the timing between that and an influenza infection, you can actually protect from influenza. And we've seen that also with some viral co-infections with influenza. If you give one virus first and then give influenza, you can actually reduce the severity of that infection. And in this particular study, they used semi-lethal doses of both pneumococcus and flu. So each one of them on their own had some lethality. When they were given together at the exact same time, that increased the lethality just a little bit. But the really robust interaction was actually when you gave influenza first and 7 days later gave pneumococcus. And that survival that does not happen. It's a completely lethal infection, and it's lethal within 1 to 2 days after the infection. And you can see that here on these images on the bottom, which are using bioluminescent bacteria, so we can actually visualize where the bacteria are. Of course, it's not sensitive to capture at some level. So at 24 hours post the bacteria, you do start to see mice developing pretty severe pneumonia. That's a pretty good chunk of the lung that's already infected. All of the mice actually had quite a bit of bacteria in their lungs. And then you can really see it at 48 hours where you get widespread pneumonia, widespread bacterial growth on both sides of the lungs, and the animals succumb to the infection shortly after. So the typical course of an influenza pneumococcal co-infection, as I said earlier, the maximal synergy actually happens at day 7. And that's not to say that it's not lethal at other time points. In this particular study, it's also lethal at a day 3 co-infection or a day 5 co-infection, but the mean time to survival, which is this line here, it changes. So if you give it earlier on, you have several more days to potentially survive, get treatment, have some kind of intervention happen. But at day 7, you're left with almost no chance for that and a really rapid decline. One of the first studies that I did with this was actually just get some data surrounding this to understand what was happening so that we could try to go in and hit it with a model. And what we found here is that we have an influenza infection that we've heard about the last several weeks. In that virus exponentially replicates, it hits a peak, it maybe starts to decline. But at this day 7 point, when we hit it with bacteria, we actually get a rebound in the virus. So the virus gets some kind of advantage where it can either replicate more or enter new areas of the lung, and you get a little bump in the virus that does start to come down and probably would clear given sufficient time if the animal would be able to stay alive. But unfortunately, the bacteria for low doses, low enough doses that if there weren't a virus there, they would completely clear the infection. You would recover no bacteria, even after probably a couple hours after the infection. But for the co-infection, when the virus had been there, you get rapid bacterial growth. And within 1 day, you've already essentially hit a maximum carrying capacity. That if you do have mice that live out to this, out for a couple of days, they will just remain at a really high bacterial load and a significant amount of inflammation. So to make a long story short, there has been, probably 20 years worth of work on studying co-infections. We have a lot of information. The short story is it's incredibly complicated, and there are many mechanisms. Some of these are just the simple fact that virus dissemination increases. There's increased bacterial attachment to cells. There's an increase in receptor upregulation, downregulation of AMPs. There's obviously changes to mechanical clearance as your ciliated cells are getting destroyed. Cytokines, depending on the cytokine, they're either up or down. Several different cell types are actually impaired. So neutrophils are impaired, NK cells are impaired. Macrophages are impaired. CD8 T cells decline, so they're actually killed off right after the co-infection. TLRs are impacted. And so this makes trying to understand what is actually happening and trying to understand the regulation of it extremely complicated. And that's where I had originally started in wanting to look at this and look at it mathematically, because we can start to tease apart that complexity and understand what the contribution is of different mechanisms, which are contributions from the virus, but which are contributions from the bacteria. How are they actually related? It might be that most of these mechanisms are actually related to each other and just downstream of one another. They might be synergistic versus additive. And then we're trying to also understand what is driving that pathogen growth versus inflammation versus disease, which are not, although they're related, they are not identically. Okay, so I'm gonna kind of take you through this a little bit quickly, not to spend too much time on the influenza model. We heard quite a bit about it a couple of weeks ago from Alan Perelson and Fred Adler, and this work was actually done with both of them. So we first started with the influenza model, and this is the model that Alan had introduced, where we have a target cell limited model that they get infected. Those cells go on to produce virus. That virus is cleared, the infected cells are cleared, and we get clearance of this virus infection. Bacteria, on the other hand, are much more complicated to model. They don't need cells necessarily to reproduce. They can, of course, do that on their own. And so their dynamics are much more highly regulated by the host response. And this is a full bacteria model. It includes alveolar macrophages, neutrophils, inflammatory macrophages, damage, and cytokines. And what we were able to do with that was actually get the dynamics of different doses. So at high doses, you actually don't get clearance. At intermediate doses, you will after a couple of days, and at low doses, you don't establish an infection. And so this model was able to capture all of that with just changing the initial condition. But for the purposes of the co-infection, we decided to tone that down a little bit and just take the initial part of the infection, which is the alveolar macrophages, and try to just look at the timing of this and whether or not just this single population could really give us some insight into the data and be able to fit that co-infection data. And so you can see here, when those other mechanisms are taken out of there, you either get immediate growth, which maybe has a small delay for some doses, or you get clearance here. And so we took both of these two models and tried to look at which of the processes in both of these pathogens were impacted by the other. And so to make a long story short, we tested several different hypotheses, and we ultimately landed on two completely independent mechanisms that dominated the dynamics. And what those two was that the alveolar macrophage clearance of bacteria was decreased by about 85 to 90%. And then the virus production was increased, and it was a bacterial mediated increase. And so those two mechanisms were sufficient to fit this data, and we get a pretty good fit to these data. I wanted to show just the parameter values of how we actually came to that. We take a lot of care in looking at our parameter values, their correlations, and this is what really gave us the insight they were completely independent. So we actually had two other effects in there. They were just really minor effects. Their values were extremely small when they estimated. This is the 85 to 90% of the alveolar macrophage depletion. We, of course, have correlations of parameters that we don't really pay too much attention to when they fall in the same function. We expect those to be correlated. That same thing is true for this other nonlinear function where the exponent and the rate are correlated. But the way that we knew that they were independent is those two processes had no correlations with each other. So since then, both of these predictions have been validated, some more directly than others, but there have been a couple papers that have shown increased virus production with bacteria. This particular one used Staph aureus. Staph gets inside of a cell, downregulates interferon signaling, that sort of lifts the suppression on virus production, and you get, of course, more virus production. There's been a couple of other papers looking at neuraminidase. The pneumococcus has 3 different types of neuraminidase. Neuraminidase helps the virus released from an infected cell, and it can actually rescue that interaction and rescue the ability of a virus to secrete from the cell when there's a neuraminidase inhibitor altering that and suppressing the influenza. So the bacterial neuraminidase can completely rescue that replication. And then more recently, there's been some work showing that there's actually direct interactions where influenza can directly attach to the bacteria, and it can do this on not just pneumococcus, but a handful of other bacteria as well. And you can imagine that if the virus can sort of hitch a ride, it can make it to other parts of the lung and of course replicate there where it might not have been beforehand. And with the bacterial clearance aspect of it, we had said that the clearance was decreased by 85 to 94%. We didn't say how that was, and our model couldn't determine how that was. But another one of my mentors, John McCullers' lab, showed that the alveolar macrophages are actually depleted throughout the course of an influenza infection, and that's what facilitates susceptibility to bacterial infections. And that's this data here. They used a phagocytic cell labeling dye to track these cells that allowed them to actually develop a appropriate flow gating for alveolar macrophages. These are what the dynamics look like, where they kind of drop throughout the course of an influenza infection. Once virus is cleared, then they will come back up. But at day 7, that exact value that we had, where we suggested it was 85 to 90%, was exactly that. So this data actually validated not only the mechanism, but also the parameter value that we had. And this was even in the face of having a correlated parameter value. So we did a little bit more work now knowing what the mechanism actually was and what our model was actually reflecting biologically. And with the analysis of the model, we sort of drew out this bifurcation, which is essentially just the unstable equilibria. So if you have combinations of depletion and bacteria that are above it, the bacteria is going to grow, and if you're below it, it's going to decay, just like in this figure here. So we went back into the lab and tested whether or not this was true and whether or not we actually had this curve right. And we did a handful of experiments. This is showing one of them where we took the same amount of depletion, but at different doses. And you can see within 4 hours, this is a really sort of short mechanism, but within 4 hours, if you're below it, you get clearance. You can actually get complete clearance if it's sufficiently low. And when it's a little bit closer to that line, of course, you don't get quite as much clearance. As you move further and further up, you get faster and faster and faster growth. And that's not terribly surprising. This is exactly what we think of alveolar macrophages and their importance in clearing bacteria. One of the other experiments that we did is what the model would suggest, and what this diagram would suggest, is that above 80%, you could literally go in with one single bacteria and get growth. And so we tried to do that. Obviously getting, being sure that you got a single bacteria into an animal is quite impossible, but we did try it with one, and then at least in one of our animals, we still recovered one bacteria 4 hours later. But at just 10 CFU, within 4 hours, you actually get complete outgrowth. So it probably is pretty accurate that you can go down with extremely low doses and get rapid bacterial outgrowth once you've hit that level. And this just shows that we did it for several other time points, but also other areas along this curve. So all these numbers on here are values of the day of infection. And we did a handful that were below this threshold, a handful that were above, and the behavior was completely predictable for all of them. When you were below, within 4 hours, you got a decrease in bacteria. If the alveolar macrophages don't clear it in that time, they will rebound. And so you get a lot of heterogeneity in the data at 24 hours. Some mice had fully cleared it, whereas other ones are sort of on this upward trajectory. And that's comparative to the ones that are above this threshold, where you get complete outgrowth and significantly less heterogeneity. And if we just plot the heterogeneity of that, this is true biological heterogeneity. It's not experimental heterogeneity. And that was something I thought was really interesting that came out of this as well. So below that threshold, you have quite a bit of standard deviation in the data compared to those doses that were above. And so we gained quite a bit of information from doing this and having ourselves and others validate these experiments, but we also still don't quite get everything, right? So we did get the process that was establishing the secondary bacterial infection, but we couldn't originally get the exact mechanism. Similarly, we got the amount of depletion, but it was a static parameter value, and we used a functional form that ultimately ended up not being correct. And so it was a nice approximation for that time point, but doesn't quite capture the entire dynamics of that population. We did get the dependency on the virus. We also got the process that was driving the viral rebound, but not quite the exact mechanism. And we also did get the independency of the mechanism. So I wanted just to make a quick point that validating predictions is not equivalent to validating models. And that probably goes a little bit without saying that we are using models to approximate. And so of course they're going to break down. Hopefully they do break down. That's where the interesting science, I think, is. But the important part is we can still make valid predictions with these models. But having that extra experimental data sort of tells us what our limits are on that. And we've done a handful of other studies with co-infection. I don't have a lot of time today to talk about these. We've done a little bit in extending the work I just talked about to look at therapies, both antivirals, antibiotics, and immune modulators, and how combination therapy might be able to be leveraged to help with that. We've looked at influenza proteins. The influenza PB1F2 protein is known to be associated with higher inflammation. We've shown that expression of the 1918 PB1F2 in particular will increase the pathogenicity of both the viral infection and the secondary bacterial infection. The modeling work that we did said it was mostly what happened in the virus that initiated a different co-infection. More recently, we've looked at bacterial genetic adaptations and metabolism. And this is a really dynamic interaction where certain bacterial genes really influence the co-infection, and knocking those out can take a lethal model to a non-lethal model, which is really interesting. Some collaborative work that we did with Julia Hurwitz's lab was on vitamin A deficiency. Vitamin deficiencies can alter various aspects of both infections, but also increased the mortality from the bacterial co-infection. We did a little bit of work collaborating with Chris Lepfer on IL-1 production during co-infection, and he determined the signaling axis that was important during the co-infection for this particular cytokine. And then this last paper is a review paper, but I think an interesting one that highlights that vaccination actually has some counterintuitive dynamics that happen with both the influenza vaccine and the pneumococcal vaccine in their efficacy against co-infections. And so I will stop there and take any questions that you might have.
Great, thank you very much. That was an amazing amount of material in a very compact presentation. I'm sure there'll be questions. Please don't hesitate to speak up.
I have a question. You said something about it's hard to collect information for humans. I see what you're doing, and you need information from cells that you're collecting. However, think about in the future, you will have all those wearables that provide you a lot of information about all sorts of things. How can your models fit into those? Can you be able to detect co-infections or stuff like that from information that comes from wearables continuously?
Yeah, hopefully. There was a study that was conducted by the CDC several years ago now that involved 6 different hospitals where they were actually trying to just discover and try to find some of that information for even diagnosing the cause of pneumonia. And while that seems like something that should be somewhat straightforward, it's not. Diagnosing pneumonia and the cause of pneumonia is actually extremely complicated. And what came out of that study is that almost nothing is correlated to the particular pathogen that's actually causing it. So as the data get better and as we find potentially more biomarkers that are accessible, which I think is a second challenge, accessing things in the lung is very difficult. Accessing them in the blood is very easy, but those two things are not always correlated and known. So yes, I think the short answer is that I hope that actually gets to be the case. Clinically, we are a long ways from that.
How long? Like how many years? Decades, 2, 3?
That's a great question. No, I don't know. I don't know if I can put a time limit on it. Okay. I would say at least 5 to 10, but I think that's on the short end.
Thank you.
Other time for one or two more questions?
Very safe.
Hey, Amber, great talk. I have a question if you don't mind.
Yeah, sure.
Have you looked at whether or not particular types of lung microbiomes might increase the virulence of particular viral pathogens?
Yeah, so I haven't in particular, but I have a couple colleagues here in Memphis that have. And they've found a handful of things. One is that it absolutely does. The microbiome 100% changes the dynamics. And that's not just in the lung, in the nasopharynx itself. And there's been a couple labs that have looked at this, that the bacterial pathogens and probably actually other pathogens that are just naturally in your nasopharynx are impacting these interactions. And that, what I had mentioned earlier about some of the therapies, as well as the vaccination, with the flu mist vaccination, that actually changed the microbiome within the nasopharynx, which then actually allowed bacteria to establish slightly better. So that I think is really complicated. A second complication to that is that within some of these bacterial pathogens, and some of the work that's been done on the metabolic side of it has shown that specific bacterial genes, and particularly with some of the bacterial metabolic genes, can influence transmission when it is there. And so that's also highly correlated to what some of the microbiome that's in your nasopharynx, and then of course in the lung, it's also impactful. So.
Okay, probably time for one or two more short questions. I wish we had more time because you so many things to investigate and pursue from this. But I thought I saw somebody who was trying to ask a question.
One question.
Please go ahead.
Yes, thanks for that very interesting talk. So you talked about biological heterogeneity. Presumably these are all mice from the same strain, like Black 6 or something. And I assume they're also age matched. I was wondering, what is the age of the mice? Does it have any impact on this number 7 days or any of the parameters in your model, or has that been looked at?
That's a good question. It hasn't really been looked at in detail. We always infect our mice at 7 weeks and then co-infect. A 7-day would be essentially at an 8-week-old animal. There hasn't been a lot of work looking outside of that. There's been a little bit of aging, which of course aging changes the flu infection, which is then going to change the interactions with the co-infection. But I would say that's a little bit of somewhat of an open question. All of our mice are age matched, and then we also ensured an even distribution of weights, because their weights can influence some of this as well. If you have a mouse that is slightly on the smaller side, they're going to succumb to the co-infection much faster. And so we try to deal with the biological the experimental heterogeneity by making sure that the distributions within each of the groups are relatively consistent as well, so that when we do get out heterogeneity, we can ensure that it's biologically meaningful heterogeneity.
Right. There's also the phenomenon, on one of your papers, you talked about bacterial genetic adaptations. There's also, within the population, there's phenotypic stochasticity. So even within one infection, there could be multiple genetic variations that could impact that. Anyway, so yes, very interesting. Thanks.
Okay, time for one last quick question. If anyone, I hope people will continue to talk with Amber offline and as part of the working group. So last call on questions for now.
Just very quickly, have you already written anything that explains the relationship between the model and the wet lab science that you're doing? And I mean, I'm working on the communication dissemination subgroup, and the question is there something that we could use to explain to a freshman biology student who's totally wet lab trained how models work with wet lab? And we can follow up with that in email or something in order to move on.
Yeah, so the short answer is yes. I wrote one specific review article that looked specifically at validating predictions from models. It gives a handful of examples. I'm happy to see that more and more people are doing this. I think it's becoming somewhat, I want to say, a standard. Hopefully a standard, or will become the norm, to where we actually have a lot more back and forth with the experimental aspect. And there are a handful of people that are doing this. And I gave, within that particular publication, I gave a handful of examples, but of course there are many more, and more outside of just, I focused mostly on viruses and what we have, been able to find out on that side. So I can definitely send you that publication if you're interested. But I think that would be great to have, especially as they're younger and coming up, and with computational biology being much more integrated within biology these days.
I agree completely that this is a wonderful way to reach out and for education. Did Daniel Reeves have a question? I saw a raised hand.
It's me. I have a raised hand, but if Daniel has one, let him ask before.
No, I was clapping for Amber.
Okay.
But thank you.
Okay. Jacob, we have to move on. So
Okay, I'll ask by email. I'll send an email.
Please do.
Okay. Thank you very much, everyone. Amber, thank you. That was a wonderful talk.